everybody. I'm Jessica Young. I'm an associate professor here in the Department of Lab Medicine and Pathology, and I'm really thrilled to introduce your first speaker, Dr. Alex Mendenhall, who is also an associate professor in our department. So just to give you a little background, Alex did his BS and MS at Texas A&M University, and then went on to do his PhD at the University of North Texas. He did postdoctoral training in gerontology at the University of Colorado in Boulder with Tom Johnson, and then further postdoctoral training here at the Fred Hutch Cancer Research Center in systems biology, quantitative physiology, and also gerontology with Roger Brent. And Alex is an integral part of our Nathan Schock Center for Excellence in the Basic Biology of Aging here at UW, where he leads the Invertebrate Longevity Core, and he is one of the core instructors of our Biological Mechanisms of Healthy Aging, T32. So you probably guessed by now that Alex studies a lot in the biology of aging, and he has a longstanding interest in variation in gene expression with aging, and I believe that's what he will talk to us about today. So thank you, Alex. Thanks for kicking it off.
Thanks, Jessica. So good to see everybody today. And so today's lecture is going to be about epigenetic gambling, monoallelic expression, and human health and disease. And this lecture is a bit of a tribute to our former faculty member who recently deceased, George Martin, who was really integral in really forming my views of biology and where the intrinsic variation in a lot of biological systems comes from. And that's what we're going to talk about today. And again, I just wanted to make sure that I give credit to a former mentor, George Martin, who I met while I was in Tom Johnson's lab at UC Boulder at a Christmas party with Leonard Hayflick and Tom Johnson and George Martin. Very formative for me. And for those of you who don't know, Leonard Hayflick is who came up with the Hayflick limit of cell division, that cells can't divide forever unless they're cancerous. And George has just had too many contributions to gerontology and Alzheimer's disease to really list. But fantastic formative years and really amazing to be here today. So I wanted to thank everyone right away before I get started. So Sue Young and Brian Sands did most of the warm work today, with Brian Sands really taking the lead on the genetic screen for control of this pathway regulating monoallelic expression. We also have partnered with the McCormick Lab to help us with RNA-seq analyses. So Blaise Mariner kind of took the lead on doing analysis of human RNA-seq data from the GTEX project, which is gene by tissue expression. So it's samples, post-mortem samples of actual humans from healthy tissue right after they die. And then I wanted to give a shout out to Alexander Sasha Gimmelbrandt, who's been a leader in the field of monoallelic expression research, and who's also helped us generate some data for FiberSeq, so actual single molecule DNA sequencing on the nanopore platform. But thanks, and that's Sasha right there. So thanks to everyone. And again, this is about monoallelic expression and epigenetic gambling, and the epigenetic gambling hypothesis, the idea that biological systems are just generating noise for adaptation, was really generated by George. So monoallelic expression, what is that? So random autosomal monoallelic expression is what we're concerned with here. It happens to maybe 10% of genes in the genomes on autosomes, where it's going to set a random bias for your mom or your dad's version of a gene. And that can be incredibly consequential if they happen to have a disease allele. So this is just sort of a quick overview of that. And it's not the same as imprinting, but it is kind of like X inactivation insofar it's a random choice that can be propagated throughout a tissue. So we live in a world of uncertainty. The future is uncertain. And having this sort of random variation generated as part of a biological program creates what another scientist named Klaus Gartner called an intangible epigenomic product for adaptation. So it might not be good for the individual to sort of accidentally express nothing but a disease allele, but a lot of the things that are variably expressed are cell surface proteins and major histocompatibility complex type proteins. So the immune system really benefits a lot from this sort of random variation that makes sure that some members of the population will be fit no matter what happens. That's the idea for why this sort of thing exists. But in human health and disease, it can sort of be troublesome if you happen to be the person that got the bad allele that's expressed. So that's not great. So the main ideas here that I want to talk about today are that we don't know how monoallelic expression manifests in vivo. We have medical cases and RNA sequence data that tells us that this must be a thing, but we can't actually visualize it. We know that people have monoallelic expression prevalent enough and persistent enough in their tissues to escape disease. But we really don't know exactly how it manifests and how long it lasts for, or if things get desilenced. But they do get desilenced. We'll talk about that. So the programs that control this monoallelic expression are absolutely completely undefined. We know that it has something to do with epigenetics, so we know to look there, but there's no pathway that's been shown to control this yet. We know that there's histone markings and sometimes DNA methylation that's associated with this, but we just don't know what's controlling it. So, and then in people, we actually need to develop assessment and control technologies for endogenously silenced alleles and viruses and transposons. And we're going to get into that a little bit more later, because the idea that we can reinforce the silencing of viruses and transposons is something to consider. And the machinery we're going to find out that controls virus and transposon silencing will actually control allele silencing, but in a different way that's going to have to be considered. So these are things that we need to understand this phenomenon that's affecting human health and disease because of these disease alleles that are randomly turned on and off in ways that we don't really understand why, other than we know that it's a roll of the dice and it's not really heritable. So I want to convince you intrinsic variation in traits exists. So if you take genes by environment, and then there's always a third component for variation that's left over, for quantitative variation that's not explained by holding genes and environments as a constant. So if we take a bunch of worms or a bunch of genetically identical or isogenic, if you will, mice, genetically identical within the limits of the fidelity of polymerase, we see that isogenic worms in a swirling flask will have an order of magnitude between the first death and the last death. A bunch of isogenic C57 black 6 mice will have differences in the first death and the last death, despite being in the same cages. And human twins, Danish monozygotic female twins, will have an average of, I think, 7 years difference in lifespan between them. And those are quantitative traits. Those are traits controlled. Lifespan is a quantitative trait, a trait controlled by many different genes. Then there are things, discrete traits. So things controlled by one gene. Many of you that study genetics know that there are mutants that are incompletely penetrant. If I give worms a KRAS gain of function mutation that causes patients to have cancer in humans, worms, even though they have a fixed mitotic lineage, will get developmental tumors in response to an EGF signal. But the important thing is, even though they're isogenic, this is what a wild type worm vulva kind of looks like. It's just sort of this, there's no protrusion on the surface here. Some animals have the same flat ventral surface with a normally developed vulva, and other animals end up having these tumors hanging off of them. And you can see the green here are little nuclei inside of cells. So you can see that some animals have like a little protrusion here, a little protrusion here. There's many cells in some tumors. There's only 3 or 4 in others. It's variable. Yet these animals were isogenic, and we can raise them inside of a swirling flask where they're just swirling around. There's almost no environmental heterogeneity, and this still comes out. And over, what we see is that this is differences in gene expression that correlate with this, with these differences in phenotype. And I'll prove it to you with human genetic cases in just a second. But the intrinsic variation in gene expression that's causing intrinsic variation in traits can be, and often is, caused by this mode of regulation called monoallelic expression. And it's kind of a continuum. Sometimes it can be 100% bias, sometimes it's only 80 or 90, but we know it's consequential for humans. And this is the same graph I'm showing you here, that's sort of the landscape of monoallelic expression. And this is actually work that Sasha did while he was a postdoc in Andrew Chess's lab. And Andrew Chess is someone that, of course, George Martin, the guy in the first slide, who's focused on epigenetic gambling, he turned me on to this person studying monoallelic expression, because ApoE4 and Alzheimer's risk allele is expressed in a monoallelic fashion sometimes. So even though you have a disease allele, a lot of times we get a disease allele and it says you have a 30% risk of this. Well, what does that mean? Am I going to get it or not? Well, these risks, these percentages may often be whether or not you silence that allele or an adjunct allele that potentiates some other disease allele, like a chaperone. Now, what these two pictures are showing here is a DAPI stain for all the DNA in the nucleus. And then you have a gene-specific DNA probe, and you can see that there's two copies of that DNA, right? It's a diploid cell, so you see one copy of EBF, another copy of EBF, one copy of DAPK1, an oncogene, and another copy of DAPK1. And then you see that there's only RNA that's read on one of those two. So this is in vivo actual monoallelic expression at the RNA level through in situs. So just convincing you that this is something that really happens in people and it's really consequential. And here it is in human disease. So the FOXP2 gene, some of you may have heard of, it was pretty popular a while ago because it can cause severe speech language pathologies if it's expressed. It's kind of an ancient gene that people thought was, I think it's related, it's from Neanderthals. So there's an old Neanderthal version of it that's apparently not good to express because you have very poor speech with this. My vague understanding, but the point is it's disease manifestation based on whether or not you express the bad allele of a gene when you have both copies. The same has happened for PIT1. It's a pituitary gland transcription factor important for development of the pituitary gland. If you're heterozygous for this and you have a bad copy, you're going to get disease. But you could also be heterozygous for this and not get disease if you just don't express the bad copy. Okay? So this is really, really important and it's affecting people's lives. And it's not just genetic disease, there's chronic diseases too. Like here's two cases of allele-specific expression for Parkinson's risk alleles. So chronic disease too. And again, right here, there's different ways to study monoallelic expression. And I'm showing you this because the medical cases and these are things that give us hope that this is a really real phenomenon because it's affecting people's lives in terms of whether or not they get not just a genetic disease, but these chronic diseases. Sometimes monoallelic expression can be prognostic for cancer. So when you get a tumor, you can have your tumor sequenced for expression, not just for genotype. And monoallelic expression in many tumors is associated with really, really good or really, really bad outcome, depending on which gene is expressed in a monoallelic fashion and in which tumor. So it's all very case specific. But the ways that you can study this are sequencing-based approaches, looking at medical cases where you take a cheek swab from a patient and see if you get the proband from the disease allele or not, which also suggests that these things must be propagated to be silenced throughout many tissues. But there's a lot that we kind of need animal models for. But what we can learn, again, in getting at the main ideas, we don't know how monoallelic expression manifests in vivo. So we can use a sequencing-based approach to look at tissues in human people. So we can analyze the GTEX data set that's available that NIH put a lot of money into making. So it's a data set of RNA-seq. It's bulk RNA-seq from biopsy tissues from 832 donors and 54 different tissue types. And this is what we partnered up with Mark and Blaze for. So just wanted to give them credit again, because Blaze really has been doing the lead on running all of this RNA-seq data on their awesome, kind of a gaming computer, but it's really, really just a really beefy computer for doing a lot of RNA-seq processing, even though it could be really fun for games. What they did was they looked at this, and you might have read in the abstract, it looks like the average bear, the average one of us, has about 1,500 genes with significant monoallelic expression going on in us. That's just looking across all tissues. The average human has probably about 1,500 genes that are really, really biased out of about 3,500 that could be. And what's interesting here is that, like lots of different aging studies have shown, that there's a general desilencing with age. And that's kind of what we expected to see, is that monoallelic expression would decrease with age. We're letting go, we're desilencing genes, we're desilencing viruses and transposons. And all of this is causing general genome instability, at least from the transposon and virus particle releases. It's causing physiological instability from the change in physiology that you're having from now expressing more MHC genes or from expressing some bad disease allele that just popped up. And that reminds me to mention our work with Raj Kapoor. There's a lot of diseases where heterozygotes have either never get it or have variable onset. And ACT-G2 related visceral myopathy is a problem where people have a dominant negative version of actin that's expressed in their intestines. Heterozygotes may be escaping this sometimes, but many heterozygotes that just have one bad copy don't have anything wrong with them till they're 30 or 40. And then all of a sudden they start having problems. And this is all consistent with a desilencing. But we don't know this for sure. This is something that Raj and I are actively investigating and trying to find ways to make different patient variants of this livable. So what they often do is they just transect out part of the intestine, put it back together, and then the patient's fine. Which, how could that be unless there was a genetic mosaic? You just can't express bad actin and be okay with it. There's no way to just have a bad actin because it makes the chain fall apart. So this is that's decreasing allele bias. But then we were really surprised to find that in the esophagus and the uterus and actually some other reproductive tissues, we actually see an increase in allele bias with age. And we'll see that this might make sense given that viral surveillance programs might be using the same end gene to actually do the silencing. So in hyperactivating viral silencing programs because of environmental exposures to different viruses, this is just speculative. We think that some of these tissues might be doing more monoallelic expression collaterally just from having increases in activity, or they might be just more mitotically active tissues that do this. We're not really sure why, but that's just sort of the phenomenology of this right now. So, and again, this is something I was speaking with a little bit before we got started, is that 200 genes have incredibly reliable allele bias over and over. And these are like MHC type genes, antigen related genes, and things actually controlling the regulation of gene expression of epigenetic genes. So they themselves will sometimes go monoallelic, which probably creates a interesting feedback loops that creates more intrinsic variation in biological systems just by messing with their own genome settings. Okay, so we know about 200 genes are consistently monoallelic across lots of people so far. Those seem like things we might want to pay attention to for human health and disease. 1,500 is too much, maybe 200 is a good start. Inflammaging from desilencing of MHC complexes with age is something that Mark and Blaise raised. It's interesting that inflammaging, which some of us are really into, may actually be influenced by some of these age-related desilencing events that are now causing immune problems by making us more immunoreactive with ourselves. So we're studying monoallelic expression. I mentioned these in vivo animal models. What could we learn if we could actually see allele expression bias happening in vivo? Well, the main idea is that we don't know how monoallelic expression manifests in vivo, so that would be good if we could see that. We don't have any genetic control of this yet. We have no idea what programs are controlling this in any systems. And we also need to better learn how we can assess this and develop control technologies. And, a nice animal model where we could actually just see it would be great. And in fact, they did it in yeast, so we decided to do it. And the next model organism that we could actually just directly see through, which is, where is it? That we could learn about this in vivo manifestation of monoallelic expression in an animal model. And that's what we did. So we developed C. elegans as an animal model, because now with precision genome editing, we can actually edit the exact same nucleotides and put in one GFP version of an allele and one cherry version of an allele, or BFP and Neptune. We've got lots of different color combinations that we can use to look at lots of different kinds of animals. But the reason we care about the worm is it's transparent. We can do reverse genetics. We can do molecular genetics. Molecular biology and omics are well developed in this system. And critically, it has a fixed mitotic lineage. So when we see an expression pattern in an adult animal, we know exactly what happened in terms of cell divisions to get there. And that's going to be really important later for figuring out things like initiation, propagation, and maintenance of these marks and of the silencing. So, and also in worm, they're tiny, so we can correlate the variation in gene expression with variation in traits pretty easily in ways that would be really difficult to do in other systems or just really costly. So this is just kind of a fun slide I made for the tools we use. This is my worm bow. So this is the worm colors that we've developed. So we tried out different fluorescent proteins that were monomeric, so they don't dimerize and we get weird effects. We do a lot of micro injection. So to edit C. elegans genome, you literally inject a CRISPR cocktail right into its gonad. And then that edits the germ cells so that the next generation, after you inject them into the gonad, then you screen on the stereoscope, and then you can put them on the confocal or on the fluorescent worm bot to sort of see what they look like. So you screen them, you find some red worms, you find some green worms, and then you breed them together, and then you maintain hets from hets. So the other thing that's terrible about studying monoallelic expression is that you have to maintain heterozygotes. And everyone knows you never want that in laboratory research. You want, isogenic inbred animals for genetics. But now, which has been just crazy, we have to maintain heterozygotes to see our phenotype, even though we're doing mutants. So forward genetics would have been terrible for this. So reverse genetics it is. So we can directly see it. And this is what George Martin would have called variegated monoallelic expression. So what it is you see some cells that are red, some cells that are green, and some cells that are yellow because they're expressing both alleles. And these are HSP90 reporter alleles in a worm. That's what I'm showing you. These are all hermaphrodites that have one copy green and one copy red. And you can see that sometimes, and this is like a red-green merge. So here's the red channel, here's the green channel. And you notice this is a muscle cell that has 0 RFP in it, but it's all red up here. And these two have 0 or 0 GFP. Yeah, these have 0 GFP. Yeah, and they're or 0 red signal in the red channel, and they're all green up here. So it's fascinating, it's striking. So you can actually see how this manifests. This is what you see if you look at 40 animals. If you start looking at 100 or 1,000 animals, then you'll start seeing some real weird patterns come up naturally in just ad libitum wild type conditions. But this is what you can see looking around. You can look up here and you can see the muscle cells are frequently red or green. You can see that, an excretory canal cell right here that's all red. Here's the same cell in another animal that's all green. And then you can see a nerve cord that's all red. So just a lot of variegation happening normally for this particular reporter gene. There's other genes that don't have any monoallelic expression, as we would expect. But this is a good one because it goes monoallelic sometimes. So now we can screen for changes in this, right? So we can see this directly. And because of this, we can also do molecular genetics and C. elegans. So we can hold locus as a constant and change the promoter or change the coding sequence or take the exact same gene and move it to a different place in the genome. And that allowed us to uncover a new role for introns. So introns prevent stochastic silencing. They prevent this random silencing from happening. So genes that are intended to go monoallelic actually have lost introns. So there's a bunch of genes that go monoallelic. So odorant receptors that were mentioned in that slide that I didn't really highlight too much are obligate, monoallelic, monogenic, and they have really, really lost their introns. They're really overrepresented for that. So that's something that we thought was really cool that we found out kind of a new role for introns. And then in doing this molecular biology, we could just say, okay, what's the intrinsic noise is the quantitative measure of this, of how biased it is. And I'll show you scatter plots of this. So the intrinsic noise is the quantitative measure of how biased things are. And you can see that in muscle cells that are diploid, they're much more monoallelic and variable than polyploid and piston cells. You would kind of expect that. Depending on which promoter you put in front of fluorescent proteins, you'll get differences in the intrinsic noise. And if you move the exact same gene to a different locus in the genome, this is an interesting thing because these are called safe harbor sites that you've heard of for mammalian research. These sites are expression permissive safe harbor sites in the worm. But if you look at them in 2 different colors, you can find that 2 different safe harbor sites have very different noise levels. So that's just something to consider, that you could take the exact same gene, move it to a different place in the genome, and it might be wildly more monoallelic. And this is something that we want to think about for cancer biology, since there's a lot of translocations, a lot of the genomic environment of different genes gets disrupted and changed. So it might change their behavior too, in terms of monoallelic expression. So we're going through our animal model. We just got a little look at manifestation of monoallelic expression and some cis elements that can control monoallelic expression. Now we can start to screen for genes using our animal model system. So genetic controls and genetic tools in an animal. So what we did here was we designed a genetic screen to do, we targeted it for about 80 or 50, anyway, a few dozen chromatin modifying genes. And again, we have our HSP90 reporters. And what this is showing you is that each time you have heterozygotes lay, lay eggs, you see quantitative population genetics really works out. They produce 25% homozygous green and 25% homozygous red. And all you want are the biallelic animals so that you can screen them for changes. So you have to keep constantly enriching for hets, but in another system, you would have to keep breeding, and then you'd have to deal with the breeding effects and things like that, which there are. So we keep propagating these animals, and we pick heterozygous animals, have them lay eggs on what's called RNAi food. So for those of you that don't know, C. elegans can just be fed bacteria that express RNA that then knocks out a gene or knocks down that gene. So you can screen genes by feeding these worms. So you feed the worms a food, and then it lets you test the function of a gene. So we feed these worms these foods that knock out 50 or 80 genes that affect chromatin. So like histone methyltransferases, small RNAs, DNA methyltransferases. So we're trying all this stuff out, and we get real weird results. Nothing worked except one gene called MET2, which is SETDB1. And SETDB1 is known for regulating heterochromatin. So I was like, all right, that makes sense. But then I was like, wait, this doesn't make sense, because SETDB1 works together with another gene to make H3K9. So getting rid of it, like, maybe that was right. So we got rid of it, and we got a lot more monoallelic expression. So we're like, okay, so now we're not doing silencing. So, okay, this kind of makes sense, but it kind of didn't. So we have this, and you can see in the scatter plot from just this RNAi. So you see that when we normally get these animals, they're kind of yellow, right? So this is one of them slapped into the tube. I showed you when we got 40 animals, the funkier guys that we saw with reddish and greenish cells. But, you know, on average, when you just slap a worm in the tube here, you're going to see kind of a yellow worm. And then with sometimes you see a little bias like we did here, and you see a lot in the diploid cells, like this is a green muscle cell. So again, like we showed you, in even under ad libitum conditions, you get a little bit of monoallelic expression. But we knocked out MET2, which is SETDB1 in mammals, known controller of heterochromatin, and H3K9 trimethylation. And all of a sudden we get worms that are green and red and yellow and red. And we know that these, and we probably got animals that were completely red and completely green, but we don't know because we can't tell that they're heterozygous or not unless we genotype them. But so we just have to look for animals that have a little bit of yellow or red so that we know that we're actually getting a heterozygote. And then later we can even do experiments to show that no, they didn't lose the gene or anything like that. This wasn't like DNA loss. They'll actually have progeny that are all red and green. So this is really cool. So this is the spectrum of phenotypes that comes out, because you see a scatter plot and you don't know if those cells, like this cell could be from an animal that was red and green. So you can see over here that the way that this is manifesting in tissues, this is how it actually manifests, is you see entirely monoallelic tissues. So that's important. So you see propagation of this monoallelic event throughout the tissue, which is now makes more sense for how someone could escape genetic disease if they just have this thing silenced throughout in the tissue, versus a variegated silencing where if half the cells express it, then it might average out to being okay. But if it's completely monoallelic throughout a tissue, then you've silenced the bad allele, or you silenced the good allele too. Sometimes it's bad when you do the silencing. And again, so what you're looking at here, this is just, so, you know, this is like a worm torso shot. So all these little tubes here, these are worms in these microfluidics. And this is just what fits in the microscope field of view, is just sort of their torso. So we show you a bunch of these torso shots over and over so that you can see that things are going from yellow to green and red and yellow. But I don't want to show you just microscope data. So we show, here's the actual cells plotted as their allele values, or the intrinsic noise quantified. So this is the kind of data that I'm about to be showing you to define this pathway. So we did the reverse genetic screen. We did RNAi, then we made a knockout. So MET2 also controls germline mortality. That's an interesting phenotype for the same gene. If you knock it out, the worms will become sterile in about 30 generations. So not only is this regulating heterochromatin, the same gene is regulating monoallelic expression and the mortality of the germline. So we made a fresh knockout allele, and we see the same kind of thing. Here we're getting, this is just one animal that's variegated, right? So it's green and red, and then some yellow too up here. So you get you get a full spectrum of phenotypes from this, because it's sort of a probability-based event. If it happens to start a little earlier, so it happens in the E lineage early, you might get the E lineage in the worm is one cell that turns into 20 cells to make an intestine. That's why we care about this, is because we can track the variation from the mitotic events where they were initiated in. So to have, because it's mathematically impossible to get a cell where all the nuclei just happen to choose green in 20 different cells. Because the other thing I'll tell you about these cells is that they have 32 diploid copies of the genome per cell. So they're not just flipping a coin. To choose all green had to happen and be initiated and propagated early because they're polyploid cells. That's another advantage or strength of this system, is it's not just coin flip statistics that's happening in this particular tissue. It's got to be initiated and propagated early, because you do see that sort of thing happening in the muscle cells that are diploid, just red and green and red and green. But for something that has 16 copies in 2 nuclei, 16 diploid copies inside of 2 different nuclei, for them to all be red is just incredibly improbable, or green. So we made these knockout alleles, then we made it with the full length HSP90 fusion protein. So it's HSP90. So we added coding sequence. We still see the same thing. And then we moved it to another, because we showed you the locus effects. Then we move it to another chromosome and we say, does the regulation still work on a different chromosome? Yes, it does. So SETDB1 seems to be negatively regulating monoallelic expression in a worm. That's a win. Now we got a gene. Now we can start to make a pathway. So SETDB1 actually is known to interact with several other proteins. So now we can just go through that usual suspects list. RL14 and Lin65 are helpers of SETDB1. So they actually sort of phenocopy it. KEC4 and LEM2 regulate docking for heterochromatin formation with SETDB1, and they have no effect. We didn't detect any effect for KEC4 and LEM2, so they're distinct from the heterochromatin H3K9 trimethylation pathway. So the big deal here to bring you into the sort of technical fold here is that SETDB1 is known to work with another gene, SUB39 or SET25 in the worm, cooperatively to add methyl groups to H3K9. So normally MET2 in heterochromatin adds methyl 1 and methyl 2 to H3K9 to histone 3 lysine 9, and then that gets finished up by SET25. So what we expected when we go down here to SET25 was to see the exact same thing we saw with Lin65 and MET2. But instead, SET25 has the opposite phenotype. We get rid of SET25 and it's SET25 is doing all of the silencing. And that is kind of what we expected. So we should see SET25. So it's weird. That's why I was confused when MET2, like I thought MET2 shouldn't be a negative regulator. I thought MET2 should be working to do the silencing. So when we got rid of MET2 and saw that everything was getting silenced more, that's why it was crazy to me. I just sort of, so seeing SET25 have this phenotype, okay, it's definitely doing the silencing. So we get rid of SET25 and we actually see that from the scatter plot that even under normal conditions, there's the normal amount of silencing that we were seeing is actually dependent on SET25. And then SET25 has a helper that helps it do nuclear localization called Lin61. So we knock out Lin61 and it phenocopies SET25. But again, we're not sure that the exacerbated silencing we're seeing in a MET2 background is because of SET25 till we do the double mutant. And that's what we do. So we get the MET2 knockout and then we RNAi SET25 and it reverts back. So the crazy variegated expression and monoallelic expression that happens whenever MET2 SETDB1 gets knocked out, that's all because of SET25 SUB39. And so SUB39 silences things. So now we're going to trip over to cancer biology. So immune escape is a big deal in cancer biology. And cancer cell lines can escape immune detection. But if you give them SUB39 inhibitors, then their immunogenicity is restored. So SETDB1 in cancers, when it's mutated, causes immune escape. And what we know in a worm is you get a lot more monoallelic expression. And what we know from GTEX sequence data is that one of the things that goes monoallelic a lot is MHC complexes, or they get desilenced. So tumors may be silencing more of these MHC complexes to escape disease whenever they knock out SETDB1. So that's one of the things that I think is important to think about that we learn from this. And this is all needs more verification. This is all coming from our toy model system, the worm. But it's the best thing we got to understand how this stuff is regulated, because it's the only animal model, and we're going to want to go and put these into mice eventually. We're going to have to make fluorescent red and green mice, but we don't want to just start trying that ad hoc. We can do that in a worm and try out what does and doesn't work, but who wants to breed together mice and then see if can we even see the fluorescence? So it has to be a very careful, targeted approach in the mouse system once we get things established in a worm or some other easier systems. So what this allows us to do is that we've now got a rudimentary 5-gene genetic pathway with a tiny arrow for RL14's tiny effect. So RL14 was kind of a wimpy effect, but it still was, we detected it. It's just a little bit of a helper for MET2, but it doesn't seem to be helping that much. So these proteins, now we're going to what part of the protein matters. So these proteins are histone methyltransferases, and we did something that we thought was really smart, but now it's not that smart because we need to mutate other domains. But it turns out you can set DB1 is here with its mammalian, with its worm homolog MET2, and you can see that the domains, the set domains of these proteins are really, really, really similar. They're really conserved. And Susan Gasser showed that you can just mutate this cysteine to an alanine and completely remove the methyltransferase activity. So to test out if that's what was going on, even though we didn't really think that was what was going on, we mutated these set domain residues. And in fact, the set domain residues are required. So just changing it from a cysteine to an alanine gets rid of all of that activity. So the whole protein's intact. It's this big old blob floating around, and it's just got one amino acid changed. Pretty convincing. Maybe it's the set domains, but what we didn't do in retrospect, we didn't mutate any other domain and say that that doesn't matter. So we have a little bit more. So the set domain absolutely required, but it might be based, especially based on what's happening with set DB1 in humans and cancer, that if we mutate any part of this protein, then it's going to be messed up if it's acting like some kind of repressive scaffold. So we have a genetic description. MET2 is a negative regulator of monoallelic expression, and set25 is a positive regulator. It's required for monoallelic expression. And the set domains are actually required. And just mutating one amino acid recapitulates the phenotype of the knockout. So it's really kind of convincing. And I really, really want to believe it. But then that puts us in a weird competitive histone methyltransferase model where set DB1 and SUV39 function differently in euchromatin than they do in heterochromatin, or they might not even be doing the same thing that we think they are. They might just be acting as transcriptional repressors and a repressor. They might not do, because in monoallelic expression in humans, a lot of times the H3K9 trimethylation mark is not informative. They can't find genes by looking for that. But you can find genes that are monoallelic by looking at H3K27 trimethylation. So maybe that's the mark, but the H3K27 trimethylation gene didn't cause this, didn't negatively regulate that. So we know that, oh, did I get to the specificity? No, I forgot that slide. So there's also the specificity. So it regulates monoallelic expression for a few different genes, but not for all of them. So there's other genes that go monoallelic that MET2 doesn't regulate. So there's multiple monoallelic expression regulation pathways. So they're undefined. We got that when we can also get at developmental control. So we wanted to get at the developmental and genetic control. So what this is, because of the propagation throughout an entire organ, which would have had to happen early in development, Brian came up with this cool experiment to just do the reciprocal crosses. And it turns out, and these are recessive mutations, so they shouldn't have an effect in a heterozygote. But what this shows is that you absolutely need mom's protein to regulate monoallelic expression. So we did these crosses with wild type animals and MET2 animals, MET2 knockouts. And if mom doesn't have MET2, you're getting monoallelic expression happening, even though you are a heterozygote and you shouldn't have enhanced monoallelic expression. And all of the heterozygous children of this animal do not have monoallelic expression. Okay, so the heterozygotes don't, unless you are the first generation to be a heterozygote, because you lacked mom's maternal protein. But if you're the first generation heterozygote coming from a dad, so if that came from a dad, so just pay attention here. Oh man. Okay, oh good. Okay, so this one has the paternal MET2. So this animal got it from the paternal, and it's, it's basically wild type. So that's from the male parents. And then you see that the MET2 from the female parent, even though it's genetically heterozygous, the phenotype is as if it was a monozygotic mutant. So does anyone have any questions here? Because I want to go through this slow because it can be a little mind bending. So now we're going to do the reciprocal cross with the single mutant and the double mutant to be sure that is set 25 acting in mom, or could it be after the maternal to zygote transition? Is the negative regulator just holding on to things and then set 25 gets expressed by the embryo? No, it's maternal. It's maternal set 25 or SUV39 that's also acting in the early embryo. So both of these proteins are need to be from mom to work. So that's what this data shows right here, is that if you got set 25 from mom, even though you have your set 25, you're heterozygous, you still have the silencing. So even though you're heterozygous and you have a wild type copy of set 25, because mom had the wild type copy of set 25, she was able to silence you. Does that make sense? Okay. So you're heterozygous for set 25. You should be just fine for silencing. But you're not. You don't get to silence anything if you had set 25 from dad. So even though you're a MET2 mutant, this animal didn't silence anything because it got set 25 from dad, and it basically was a set 25 mutant from its mom. So if it doesn't have mom's set 25 protein, even though both of these animals are MET2. So the point is these animals should be really, really monoallelic. Okay. And they aren't if they got the set 25 mutation from dad. I mean, from mom. So does that make sense? I'm probably drilling down too hard on this, but that's what the reciprocal crosses tell us, is that it's the maternal proteins acting in the early embryo. And that's really important to think about for human biology, because that also shows us how things could propagate. And the other thing that we're getting out of this is spectrum phenotypes. Even though we're in the exact same mutant, some of the mutants will be variegated with red, green, and yellow cells, and some of the same progeny from the same animal will end up being all red or all green. And I'll show you some of this. This is not heritable at all. And you get a spectrum. So this is one parent we got. These are really hard, terrible experiments that Brian did, because you have to find a heterozygous parent and then let them lay eggs for 2 hours. And 50% of those are going to be animals you don't even want to see, because they're going to be all red or all green. Plus, they're already a mutant that can make animals look mostly red or mostly green. So this is a MET2 mutant that's got a really funky expression pattern. And MET2, the other reason we did this experiment is MET2 also has a permanent RNAi phenotype in the germline. So set DB1 mutants in worms, in addition to controlling the mortality of the germline and doing heterochromatin formation, they also have this permanent RNAi phenotype where they will silence a gene forever in the germline, but not in the soma. So you can see that these things are the offspring of this animal come in all different flavors of allele bias. So that's important to consider. It's not heritable. It's not transgenerational epigenetic inheritance at all. So checking the time here. All right, so this is our working models for regulation of monoallelic expression. This is our genetic pathway right here. So it's just 5 genes controlling a pathway. This gives us a handle to study things, because now when we do molecular biology and we do omics stuff, now we can have really, really enriched monoallelic expression. And this is what we think is going on. So Lin61 and Lin65 keep set 25 and MET2 in or out of the nucleus. So they help them get into the nucleus. And then RL14 helps MET2 do something to prevent set 25 from silencing genes, right? And if everything's okay and MET2 is healthy and functional and around, you'll get some transcription factor binding and expression. And if not, maybe you get an H3K something mark. We don't know what the mark is. We got to find out. And then you get silenced. So those are the main ideas. We're getting down to the last one, assessment and control technologies. So in vivo manifestation and genetic control and genetic tools are things we can use in our animal model to get at these control technologies. So that's what I'm highlighting here. And what we want to do is eventually we want to reprogram the epigenome for longevity and genome stability. We want to turn off the alleles that are shortening life, turn on the alleles that are, make sure that we're maximizing the expression of those. We can turn on genes with dCas9 with a transcriptional activator. We can turn off genes with dCas9 with a transcriptional repressor or with RNAi. So the ways that we decide to silence alleles or activate alleles may be different and have different kinds of effects on neighboring genes or different efficiencies with age. There might be something that is higher risk, but it's the only thing that works when you're 75. We don't know yet. We don't know about this, but there are 4 siRNA drugs on the market already. Epigenetic medicine is coming. We're already doing genome editing for people to curing sickle cell anemia with genome editing. So epigenome editing is coming as well. It's happening in cell culture to turn off bad TP53 alleles, and eventually this thing will probably be coming to the clinic. So what we want to do is start getting at the basic biology of collateral damage when you silence things with one technology or another, or activate things with one technology or another. What happens to adjacent alleles when they're oriented in one way or another? And we have an exciting, we're kind of nerdy, techie, exciting 4-color system. So we have these 4 spectrally distinct proteins that we can do what's called fast sequential scanning. So we can look at all 4 things in one animal with pretty good fidelity. So we can see how 2 different genes alleles are turning on or off. And what happens when we silence a gene that's normally monoallelically regulated? What happens when we activate it? What about a gene that we try to silence that's normally biallelic? What if it's next to a monoallelic gene, right? All of these things, what if it's oriented towards it? What if it's head to head or tail to tail? There are all these things that are happening in our genome that we don't know. And we're giving people siRNA drugs to turn off genes, and we don't know what's happening to the genes next to them, right? There's SUVAR is the spreading of variation in heterochromatin. So that's the old school position effect variegation. Is they just found that a gene could get turned off if it was next to another silenced gene. And we don't know what's going on at the allele level. We're in a world where people are often thinking about editing genomes for people that are homozygous for this or that, but really looking at the human GTEX data, most of us are heterozygous, especially at these alleles that go monoallelic. It's easy to find sequence heterogeneity. I thought it was going to be a problem, but it's not. So the other thing is we got criticized for this before in our proposal, is that can we even turn off an RNA? Can we use RNAi in monoallelic regulatory mutants? Could we use RNAi in a tumor that's got crazy things going on with its epigenetic regulatory systems through different mutations and whatnot? And what this slide is just us using RNAi. These are animals expressing a reporter allele. They're just in one color, and they're some of the monoallelic regulatory mutants that we identified, the set domain mutants and the knockouts. And all this is these are the worms are red. You can see in the MET2 mutants here, they're kind of a little patchier. So if you look in one color, the animals that are silencing alleles, they'll silence them if they're both red or if they're both green. They just look patchier. They kind of have a spotty expression pattern. It might be kind of hard to see in this, but the point is we can RNAi cherry. So we can knock down this gene, and then we see that the expression level actually comes back up. There's no permanent somatic RNAi. There's nothing funky going on. And actually it's refreshing and reassuring that we can operate the 2 systems independently. The endogenous silencing that's happening can be turned on and off. So we can turn off the allele, and then it actually comes back up after a few generations to its same sort of low modeled expression level in the MET2 mutants, or in the set25 mutants, or in the double mutants. So these are important little first steps that we need to sort of get at the therapeutics of this. So monoallelic expression is variably initiated in development, affecting patterns in tissues. If you get it initiated real early, you get an entirely monoallelic tissue. If you let it initiate it a little bit later, the cells will autonomously choose their alleles, and then that will be propagated throughout development. So for a more variegated tissue, as George Martin would have called it. So one controlled program is now genetically defined. There are other genes that we can see with fluorescent reporter alleles that go monoallelic that don't respond to this. So it's a very, some genes are regulated in one way monoallelically, and others are regulated a different way. And we see this in human tissues too. Some genes respond to 5-ASO, which is a DNA demethylating drug. So some genes will change their monoallelic state whenever you affect DNA methylation machinery, and other genes in humans, all the monoallelic expression has to do with just histone markings. So both things are happening in people. So there's multiple modes of regulation. It's not going to be as simple as we would like. So the new antagonistic roles for SETTB1 and SUV39 also fit cancer immunogenicity data. So that was really refreshing and reassuring to see that, like, oh, good, at least the new antagonistic directionality of these is recapitulated in some ways that make sense, so that we're not off in crazy town. Hopefully, still might be. And then RNAi can target alleles independently of this endogenous silencing machinery, which was really important for us to find. Dicer is an important component of the RNAi machinery that also didn't affect anything in our screen. So that's cool. And we don't know anything about the chromatin. Sasha is who I pointed to before. He's a sequencing maniac. He's really gotten into all the long read sequencing technology. He is 50% local. He's half-time at Harvard, half-time at Altius, and he's really, really good at doing what he says fast. Anyway, he's been fantastic to work with, and he's been studying this forever. So he was in as a postdoc in Andrew Chess's lab, getting hybridization arrays to work to show that this was widespread in 2007. Just SNP hybridization arrays before RNA-seq. He knows the best ways to get at this with RNA sequence analysis, with spike-in controls. He's gotten at this with ChIP-seq data before. He's really, really, really lucky. It's really lucky for me that he happens to be in Seattle now, and he's really excited about the animal model. So we're really excited. This is long read sequencing data from C. elegans that we've generated. And we're really excited to get some of the fiber-seq data where we can sort of see how nucleosome occupancy changes, and then start to, do the ad hoc ChIP-seq stuff with cut and tag technology from Steve Henikoff, who's also been really nice and supportive, which cut and tag is just a cheaper way to do ChIP-seq that's a little bit more targeted. So that's Sasha again. He's been great. Future directions we need to go into, more pathways, more about the chromatin biology, clinical assessments, the technologies to change these things. How do we deliver these technologies to the right tumors and right tissues? Is it going to be injection, biopsy? We don't know. Delivery technology is always a big bugaboo for genome editing. Like, where do we actually get the genes edited, and how do we deliver the right things to the right places? And now, if there's 1,500 genes that are being randomly silenced out of 3,500 that could be, if that's even right, it's a soft number right now, and we all have different genetics, no one physician is going to figure that out. We're going to need AI helpers. We're going to need AI to help us figure out what the hell to do. Like, what is our treatment plan going to be for optimal human health whenever we know that, okay, this is my expression profile, and this is how my expression profile is changing with age, right? So I think that's going to be part of the future of medicine, is having, expression profiling of you throughout time, and then saying, oh no, it looks like you really got a lot of transposons. We're going to have to do a round of therapeutic silencing. Your transposons and viruses are just getting released right now. It's had a stressful event or something, things like that. Or, you've got a risk allele that's become desilenced, or you seem kind of bloated. Let's tone down some of your MHC expression that's kind of popped back up. These are the kinds of things that we would envision for epigenetic medicine and therapeutic re-silencing of viruses and transposons, right? Just keeping that stuff silent. We don't want those jumping around and messing things up. So anyway, thank you for your attention. I know I'm a little over, and I want to address any questions I can. Sure.
Hi, thank you. Very great talk. I want to ask, so what makes the mom's copy of MAT2 and SAT25 special?
So sure. So what makes the question is, what makes mom's SET25 or MET2 special? So she's asking about the reciprocal crosses. And what that is that in the early embryos, there's something called the maternal to zygote transition. So early embryos often rely mostly on mom's proteins. There's very little zygotic transcription going on in the early embryo. So it's actually mom's proteins are being what's used in the early embryo. The early embryo doesn't even make any new proteins. So even if it has a good copy, it doesn't matter because it's using all of mom's copies. That's why those reciprocal crosses were a little bit hard to explain, maybe because I didn't, I privileged that the maternal to zygote transition was kind of known, but we often forget about that sort of thing. It's something we all hear about in biology 101 when we're freshmen, and then, oh yeah, that happened. So it's a maternal effect. This is sort of a classic maternal effect. I hope that helps. So mom's protein is special because you only use mom's protein in the early embryo. So that's why doing those crosses where the progeny should be wild type, they're genetically heterozygous. But if they didn't get mom's protein in early development, they might as well have been the mutant, because the only time that you use that protein to set the monoallelic expression up is in early development. And that's why it propagates throughout the whole tissue. So that's a really important experiment because it helps us understand how you could end up with a tissue that was all mom's version of a gene or all dad's version of a gene through random probabilistic silencing. And we see that sometimes we get all mom, sometimes we get all dad, and sometimes we get a mix of mom and dad and both in the tissue in the exact same genetic background. So there's still things we can't explain, probably because they're meant to be a probabilistic distribution. That's the point of it. I hope that helped. All right, we got a couple more questions.
Hi, is it working?
Yes.
Okay, I can't tell. So if all of that gene silencing is happening in the early embryo and you know the cell lineage, can you trace the exact point at which that decision is being made by looking at like?
Yeah, so we can actually see, because all 20 cells from C. elegans intestine are derived from one progenitor E cell, we can really see that, and we can even see whenever it looks like something happened later in the development because we know the lineage. So we can see like modeled patterns where different lineages made different choices at different points in time. But it looks like most of the time, and we do catch those animals are rare, we can see lineage specific events, but it looks like the binary thing that happens most of the time is things are usually getting set early in development, like the whole E cell goes one way or another. And then we'll see like 18 cells will be this, and then it looks like the fidelity was lost or the mark was lost. Like then 2 of the cells might be biallelic kind of randomly, like it was just a low fidelity process. And Gendrel and her student, so there's some heavy hitters in this. Gendrel, Christine Desteches in our department is buddies with, and I forget her name, but her student has a cool mouse model where they just sample the cells. And the point I'm getting at here is her framework. So one of the ways that she thinks about this is initiation, propagation, and maintenance. So you can see in the lineage that it looks like some things were failure to maintain versus some things were early initiation or failure to propagate. There's failure to propagate and failure to maintain, which you can kind of see if you get like biallelic expression or random other things. So that's how we can kind of use that. Yes, I want to make sure I get everyone's questions. Thank you. That was a great question.
Hey, Alex.
Okay, good. Sorry, thank you.
Hey, Alex. Fantastic as always. And I wonder how plastic is the system? So if you were to do a microinjection, instead of microinjecting the germline, you inject wild type MET2. Did I get the gene name right? MET2 protein in the mutant. Are you able to reverse the phenotype?
Well, the reciprocal crosses would suggest that. So that's kind of what I would imagine, just because the reciprocal crosses show that it's got to be the maternal protein. So we believe that we should be able to do that, just because I think that's exactly what the reciprocal crosses suggest.
Yeah, no, well, I want to push back on that. It's a little different. This is a adult animal where you have established now the monoallelic or like the heterozygous expression. Can you like reverse that to the opposite?
Oh, so
since the mutant is recessive, right?
So yeah, so this is in an established soma.
Yeah.
Could you desilence something or start the silence? So
Exactly.
you can always start more silencing through RNAi. I don't know if this system, I haven't been able to get to tweak the system into doing this, but we do see more endogenous silencing, and we don't know when this happens. So if you starve the animals, they go monoallelic, and it looks like they actually might be going monoallelic post-development. So like, as in like, it looks like because we accidentally starved them when they were L3s, it looked like, then we saw the animals looked a lot more monoallelic. So it looks like they might be able to shut things down in response to environmental perturbation, or we starved them earlier and we just messed up. So we could have starved them earlier and not known it. But the experiments, this happened a couple of times with different new students trying this out. And then, oh no, you can't put 500 worms on a plate, dude. That's not going to work out. Well, you've seen the power of worm consumption. They've eaten themselves out of every, Oh, wow, some few made it to adulthood. Let's take a look and see. That is weird. Huh. That's real weird. It looks like it's environmentally responsive, so we got that going for us. But we haven't really delved into that. So you really actually bring up a really good point about persistence. So we think it's persistent throughout the lifetime for some things, but for some genes, but then Raj Kapoor's work suggests that for some genes like ACTG2, some genes might get desilenced to cause disease. Like people that are heterozygous for ACTG2, they're usually good. And a lot of this, we actually think that the bad combinations of alleles just get spontaneously aborted. That's what George said, that there's actually a lot of mammalian spontaneous abortions because there's probably a lot of aneuploidy, right? There's a lot of aneuploidy that we see in embryos and spontaneous abortions because of, genetic abnormalities. So there's also probably some of that going on with epigenetic gambling. It's my speculative conjecture. But yeah, the persistence is a really important thing to think about. Like, how long do these alleles stay silenced? And I'll just switch to the Danish starvation cohort. They've realized that some of those epigenetic effects that were kind of maternal effect seem to be persistent throughout the life of those individuals. So that was one thing that I'll say, is that sometimes silencing, like imprinting, can be persistent throughout the entire life of an individual. And then sometimes it isn't. And it seems to be a gene specific, it's case by case for different genes. And the other thing that I was saying for Mark and our actual clinical friends is that when you have translocations in cancer, the same gene that was probably not very monoallelic, or maybe went monoallelic a bunch, if it goes to a different part of the genome, that behavior can change because of its local chromatin environment that we don't also understand. And you had a question?
Hello.
Anyone on Zoom too? Okay. All right, Zoom, you're welcome. We'd like to hear from you.
Hello. Is it working?
Yeah, I can.
I was just wondering, when you do the MET2 knockout or like the SET25 knockout or even the RNAi food, are you able to target a specific type of tissue, or is that just going to be a whole body effect?
So I can tell you what tissue we're not targeting. We're not getting the nerves or the pharynx because those are recalcitrant to RNAi. But all the other tissues we're definitely getting. So we're getting the germline, we're getting everything else. And the where it matters is probably the germline. So it probably matters in the germline because it's loading it up into the oocytes, and it's the oocyte set DB1 and the oocyte SUV39 or MET2 and SET25 rather, that's actually where the action is happening. But what Alessandro was mentioning that we didn't kind of get to yet is that so we haven't tried desilencing things. So Brian in my lab has done these terrible experiments where he gets the worm, puts one worm on a slide, takes an image of it, then takes the worm back off of the slide, comes back 2 days later, puts the worm on a slide again. They don't like this. Then takes the image and then gets it off the slide. Then comes back 2 days later and puts the worm on the slide, gets the image, takes it off. The allele ratios are the same. So through like middle adulthood, and maybe it changes later, but when expression is still good enough to see when an 8-day-old worm is like a 40-year-old person, maybe. Actually, that's probably not even like a 40. But anyway, they're getting up there at 8 days old. And you can see the expression pretty well. They still have, the bias is still the same, but it might change. We wanted to do things like RNAi SET25. We haven't done this. What if we get rid of SET25? So Alessandro, to your point, we haven't RNAi SET25 in adults yet, because we'd like to see, is that required to maintain the silencing? So once the silencing is initiated, if we got rid of SET25, would we see everything becoming biallelic in that generation? Or once it's set during development, is it locked in? And if it's locked in, then we're going to have to do something like the DCAS9 with an activation domain to turn things back on and off. That's why I think these activation and silencing technology experiments are going to be real important for helping us figure out what's doing people so we don't kill people accidentally. So yeah, did that? Okay, I wanted to make sure and happy to answer anything else. And I have an email. It's real easy to remember, alexworm@uw.edu. And I'll be your instructor for the rest of the semester. So I'm around. Anything else I can do to answer any questions or resolve any confusion? Any speculative conjecture? All right. Thanks, Julie.
So, and I may have missed it. So, basically, what about, so not necessarily. So first of all, the C. elegans is all post-mitotic, right? So, okay. So, and that goes to that maintenance issue, post-replicative. So I guess what I'm driving at is, do you think it might have to do with the rounds of replication when things get pre-established? And if so, then of course you wouldn't necessarily have switching in the post-mitotic tissues, or not so much. So the question is, with these kind of massive studies where you look at different organs, maybe human organs, do you have correlations with
Organ to
mitotic,
organ.
so replicative versus non-replicative tissues? That's number one. Number two, high or low expression, RNA expression level. Is there any correlation there? Or tendency to do monoallelic and all of that kind of.
The overall thought is that things that are lowly expressed tend to go monoallelic more. And I would say, I think that's a rule that kind of got established from bacteria, but we actually see that things that are highly expressed or medium expressed, I'm not sure that's a rule in metazoan systems, but it might be. So that's definitely a rule in prokaryotic systems. And I know there's titration of things too even in metazoan systems. We've seen things where we make a single copy of a transgene, and we see a certain expression level, but then we tag the native gene, and the native gene's expressed at a higher level. So it was like, wow, did we titrate things out by putting a second copy in and getting a lower expression level? I don't know. So the short answer is the rules of low expression level that we thought would work don't seem to be working that much in C. elegans, because we thought that when we had lower expression levels, we should see more monoallelic. But if you look on some of the scatter plots, even at the lower expression levels, there's biallelic, and it seems like it's scattered pretty equally. But you know, it's still early days. We still really don't know that much. We know that phenomenologically what's going on in people, and that there's medical cases where this is important, but where you can escape disease or succumb to it. But really, these animals are really helping us understand like how it does build up in a tissue. What can we do to change it? And it's a worm. Nobody cares about a worm. Nobody cares about a mouse. We're gonna have to do a mouse next, right? What happens when you have circulating factors in the blood system? And there's all kinds of things that we have to do. But I think the worm is really the sweet spot right now, because the other thing I would say to you is that in cell culture, monoallelic expression is mitotically stable forever. So that's why people were like, is it real in animals? We don't have an animal model where we can see this in. All we see is cell culture data and sequencing data and medical case data. So the yeast people in 2004 colored the alleles differently. Aaron O'Shea and Jonathan Razor. So Aaron O'Shea is like head of HHMI now. I emailed her. She doesn't care about this stuff anymore. But the point is that the yeast people sort of came up with this in vivo approach, and that was what I sort of got focused on as a graduate student. And then I went into Tom Johnson's lab and got into this variable gene expression stuff, and now I'm back onto monoallelic expression and trying to figure it out. So it's mitotically stable. It seems like it's mitotically stable throughout the development of the worm tissue, but we haven't, so it develops the tissue mitotically and then it just stops. So we just don't know what it's doing in a mouse tissue. But the esophagus and uterus data, there's also this other data I didn't show.
So there's no correlation how much replication.
Well, there might, so that's, I would say there's higher incidences and lower incidences of monoallelic expression in tissues that are more and less mitotically active. So the esophagus and the uterus had a lot more. And then they thought it was generally reproductive tissues. Oh, crap. Well, I just went through this, but I'll forward you the slides, but the uterus, the vagina, and then some other reproductive tissue will all have higher monoallelic expression with age. So does the esophagus, right? And those are all tissues that might have higher mitotic. They all have higher mitotic loads than the rest of than like the heart and the brain, which had decreases in monoallelic expression with age, which you kind of would expect. You're not a lot of mitosis in the brain. The heart, I don't know if you do a bunch of steroids maybe, but not too much. Yeah, so it's certainly something to think about and talk about. Welcome any discussions about this stuff. We need more people thinking about it, in my opinion. We're over time, so I don't want to keep you guys. There's lots of stuff to go do. It's 5:10, so enjoy the rest of your evening, everyone. Anything from Zoom? Signing off.